So it's a pleasure for me to host this workshop on barley genetic resources. As you know, there's a lot of local resources. We have been presented for those during the first days here, but there are also dedicated genetic resources assembled. And we think this will be a little bit informal, but all the speakers have prepared maybe 10 minutes presentation, and you can ask questions on the way. I'll try to keep you online because when we finish, then we have, I would like to invite you for a beer or two maybe. But before that, they would like to give you some background how you make beer. And that can take 2 hours. No, it won't, but I hope you'll enjoy it, but first we have to work a little bit more. And we'll start out with, actually there's also 2 presentations which we cannot have, and they're not in the list. One is from Canada, and the reason is Canadians are not allowed to travel abroad during the COVID, and the same is true for China. So the Chinese could not come here and present the germplasm because maybe they can get here, but it'll take a long time to get through the quarantine when they come back. But we have many presentations anyway, so let us start with Nordic Gene Bank, because we are local, and it's Jan Svensson will present the barley collection at Nordic Gene Bank. And I'll cut you off after 10 minutes. Yeah.
Okay, nice to be here. Unfortunately, I'm not here the whole meeting. The staff actually wants vacation, so I have to do the scoring in the field myself. And here we can actually see my favorite field of this year. This is one of the fields we have at Nordion. And I apologize, it's mostly oats, but if you look to the right side, you have actually barley also. And we are the Nordic Gene Bank, and we are on the top of the world. So it's kind of important that we have a gene bank for the Nordic region, because you can think that we have very special geographical location. If you look on the south of Sweden and Denmark, we are more European, but you go further north, you really need to have adapted material. And the gene bank at Nordion, we are in Alnarp in Sweden, so that's the south of Sweden, the Mediterranean part of Sweden. It's kind of hot. And we are a gene bank for all the Nordic countries, also the autonomous regions, Åland, Faroe Islands, and Åland. And we have it Alnarp, seed storage, and greenhouse, and all this that any normal regular gene bank would have. And our collection, it's not one of the biggest ones. It's about 500 different species. And we have about 33,000 accessions in gene bank. And our owners, that are the Nordic Council of Ministers, have put our ruling for what we should keep. And it should be Nordic origin or Nordic relevance. And of course, relevance for me or for someone else, so that's a bit of a fluent area. If you look on the groupings for our gene bank here, and to the left, this is my collection, the cereal collection that I'm responsible for. And we have a vegetable collection, and unfortunately, a lot of the breeding programs, or all the breeding programs for vegetables in the Nordic region are kind of not exists anymore, but a lot of material is actually kept in gene bank. And we have a forage group. And this group is probably the most traditional group where you actually have a lot of collecting done in the Nordic region. So if you're into forage, you can see. I'm not going to talk about accession 1, 2, 3, 4, so let's just have some overview. And if you just look, we have almost 17,000 Hordeum, 2,000 or 3,000 Triticum, Avena is quite small, and Cicale. So we see it's a really kind of weird balance, probably you would say, and I will explain why it looks like this. If you just look on the Hordeum, we have, of course, like any gene bank would have land races, cultivars, and wild material, but what sticks out for our collection is actually that we have a lot of breeding and research material. So you can actually see that more than half of the collection, maybe it's 60%, is actually kind of very specific material for breeding and research. And we have also some smaller special collection. I was really happy to hear today about the bulbosum integration lines from ICADA. It's a fantastic small set, and it's really fun to look on the morphology and so on also. And we have some wild pitch species and local varieties, that's like 1,300, and I've seen some talks here about Hordeum species, and of course we have a whole Hordeum taxa, and this was collected by Roland von Bottmer, that's our retired professor. So it's a pretty nice collection if you're interested in Hordeum crocodile relatives. We have here is a big chunk that kind of sets the balance of the collection is kind of tilted really strongly to this because you have a Scandinavian barley mutant collection. And it's 10,000 accessions, so it's pretty big. We also have a nearasthenic lines that probably many of you, if you work with this, are familiar with. This is derived from a mutant collection. It's about 1,000 accessions. So we have 11,000 mutant accessions. And in addition to this, we also have translocation lines and duplication lines. And I think there is actually quite a big overlap with the world collection that is held in Gattersleben. And the mutant collection, and I think one thing that's maybe different from other gene banks that have a mutant collection is that we have stored all alleles. So if you're lucky, you can actually be in and you can have 20, 30, 40, or 50 alleles for one gene. And of course, now when sequencing is getting cheaper, and if you have a hypothesis candidate genes, you can actually go do some sequencing and probably identify many of these genes, or some of these genes. If you only have one allele, of course, it's much more difficult, but if you have 20 or 40 alleles, you can actually try to do some work. And here we have Uda Lundqvist. I mean, many of you will know Uda, and this is actually the first body symposium where Uda is not with us. So it's kind of sad. She would always correct me, and she was always a super asset when it comes to identifying this. We distribute only 10 seeds because we have so many, so you have to settle. There is a database for information that Uda created, and it's nordion.org/BDS, and that's really a lot of information. Unfortunately, we have a separate database where you do your orders. So that's Nordic Baltic Gene Banks. And this international database for barley genes and genetic stocks have a lot of information. I just took some snippet for Art A, and you can actually see there is a tremendous amount of information. And there is pictures and description of the phenotypes and so on. I can recommend. In the pipeline, we have those two kind of sub-collections from mutant research. It's the chlorophyll biosynthesis for anthocyanin mutants. But these will be parked until we have some funding to actually do some work on this. And what we need to do is to link these two databases together, of course, so that you can be in the BDS and you can click and make an order or find some, go back and forth to this. Experience for these special collections, validation can be very difficult, time consuming if you have thousands of mutants. We have introduced a policy, we only take stable mutants. We have some mutants that are lethal, and we actually not continuing to work with this because it just costs us too much money to maintain this. And the mutant collection for Nordion, if you look at all the Nordion, is actually the highest number of orders. I don't remember exactly, but probably 400 or 500 every year is sent out to different research groups. And most of the collection is in pretty good status. It's taken Nordion 20 years actually to get this collection up to good status. And the remaining 3%, these are the tricky ones, of course. There's always some that have some, you can have a very nice spike, but you only have 1 grain per spike and so on. So probably these will have to be left. And what can be done on a global scale? I just had some thought when I was thinking this. I mean, there are many gene banks that have this type of collections, and maybe there should be a little coordination so we don't have overlaps. And maybe sometimes there could be some form of consolidation so we don't waste funding to keep the same type of material. And I think if you look at the Arabidopsis community, they have a really fantastic system in a sense for the special collections. I mean, you have you have NASC and you have Ohio, and then you have Riken in Japan, where they send out hundreds of thousands of samples to researchers all over the world. Another thing is that, I mean, usually when I talk to people that are kind of the owners, being politicians or this, is that gene banks, in a sense, and I don't think people always understand, but it's a biological infrastructure. You can easily get an infrastructure grant to buy a machine, but to try to explain this, but these genetic resources, it in itself is actually an infrastructure. It's a biological infrastructure. And maybe there would be some opportunities within the EU grant system to apply for some infrastructure grant to look on this kind of material is special collection, because I think most gene bank would have some type of this material to work with. I go back to my old field here. I miss it. Thank you.
And that is a presentation by Andrea Visioni, and it's from the Carter Barley collection. So we go to a different climate, or region, I guess.
Okay, I will start with the good news. We finally inaugurated our new gene bank in Rabat. It was a couple of weeks ago with the Minister of Agriculture, our DG. Behind you can see all the barley lover Ahmed Hamri. Okay, so yeah, the gene bank and the genetic resource unit, that includes also the pre-breeding, that is my actual position at Picarda, is doing basically phenotyping, genotyping of genetic resources. We do pre-breeding initiative. We share a lot of germplasm around. We are sequencing with the help of many institutions. I think the most important one probably is the APK with Dr. Neil Schlein. We are sequencing a lot of our accessions. And of course, we have contribution also in the database and all these tools that allows to select this kind of resources. Okay, in number, our collection is basically ranked as the second in the world. I don't know of how recent is this data, was given to me from my colleagues. So we have 30,000 accessions of barley and 2,300, almost 300 accessions of wild relatives. Then if you look, we have 58% of this is basically land races and other wild relatives. More in depth, you can see that we have, of course, 30,000 accessions on Oldeum vulgare and all the other genus that you can see in the table. And very important that out of the whole collection, we have 45% of unique accessions, and almost 78% are phenotypically characterized. So, of course, as international organization, we produce international public goods, and we basically share germplasm with anybody that wants to need it. And this is basically what we did, this distribution, a summary of the distribution of the last 5-6 years. So you can see our material is traveling quite a lot around the world. Of course, we are always trying to add new genetic diversity in our gene bank. So for doing that, our colleagues, they produce this gap analysis to identify the regions and the traits that are needed for our gene bank. The last 3 collecting missions were before the pandemic, of course, and were in Mauritania, Tajikistan, and Lebanon, for example. And then, of course, we are not only sharing germplasm, but also we do some tests. This is just an example of what we are doing in Rabat, in Morocco, in 2 stations. One is our main breeding station, that is Marchouch, and the other one is Sidi Al Altazi, that is a very important hotspot for disease screening. It's close to a river, it's nearby the sea on the Atlantic coast, so the conditions are very, very good for many pathogens. We also evaluate for quality traits, for instance, beta-glucan content. This is the results of some test that we did during 2 years, some time ago. Of course, then we have this fix that is basically focused identification of germ plants and links adaptive traits, environments with associated potential pressure, and the GBank sections. Basically, it's a kind of search tool to reduce the number of sections that you may need for studying a determinate trait, like such as a disease or a quality trait. Again, this is the results of another study that we did with our fix. These are for NetBlotch and Spotform on NetBlotch. And on the bottom, you can see also a study that we did for its tolerance. That is also quite important in many environments where we work. This is a collaboration we had with EPK again and with the University of Minnesota with Professor Stephenson. Basically, we selected some figs lines that were resistant to multiple disease and were sent to see the winter survival in Minnesota. And we found that many of them that basically are originally from Mediterranean region are survived pretty well in the harsh winter of Minnesota. The evolution of figs is combining this technology with machine learning. This is a recent work published by our group, basically focused on the resistance to scald. And in general, our pre-breeding activities are basically, we produce a lot of crosses, we test this, we advance the generation, and we test this material in our station. Then we do a selection with the breeders, and the breeders, they enclose this material, the most promising one, in their breeding program, either for further crosses or for just evaluation. This is a bit a summary of what we had in the field this year. We had around 1,000 accessions across different generations. And this, for instance, as you can see, the box plot show the total biomass, the grain yield, 1,000 kernel weight, number of spikes per square meter of our spontaneum derivative lines. So they are doing pretty well compared with the local checks. Then this is the same story, but into environment, during another season. Again, you can see that some of our derivatives are outperform either the elite lines of varieties and the checks. Again, here the same story for others with other spontaneum derivatives, but it's under severe drought. It probably was done during a very dry year that we had a few years ago in Morocco. This is the part of the collaboration that my colleague was citing before. We tested this. I mean, this material has been tested a lot for disease resistance, but we also wanted to test it for yield under drought in our environments, and that's what we got. So these results are quite encouraging. And this is just to show you the evaluation that we did for the resistance to powdery medium. Now I'm going to introduce you our new toy. Let's see, is a lysimeter. It's basically a fully automatic robot combined with balances, and we will measure the transpiration in a gravimetric way. We have a sensor grid installed along all those pipes, those pots. And the pots are filled according to the soil of the station where we work. So the idea is running experiment in parallel in the station and in the lysimeter. And the system is fully automated. Basically, you can choose, I will show you in in the screen, you can divide the lysimeter into different sectors. You can apply different levels of water stress. Also, we are in September, October, we will get the hydrophenotyping system that will be installed on this bridge that you can see in the picture. So moving around, we can take pictures and we can determine, we can collect data about green fraction, greenness, air count, plant biovolume, etc. And there are many other traits that still work in progress. It's kind of collaboration we will have with the company, with the supplier. And also, one of the idea is, I saw this article, and my idea is adopt the protocol that they use in the lysimeter. But in this case, we will use a mix of sand and soil, because otherwise, I'm afraid that will be too complicated to extract the roots. This is, again, a second part. I think Miguel showed it before. It's our Phenobuggy. It's a tractor-mounted visual head. So you can see that we have quite a lot of traits, like plant age, green fraction, green area index, etc. Also, more bottom, you can see plant density, steam density, air density. All this was developed in collaboration with the company, Eiffel, and most of the traits it was a big discussion with the breeders, of course, in order to choose the traits and the type of sensor and adapt it to the budget we had. And that was not really easy. And now I'm going to show you one of the collaborative projects that we have. This is the Agent project. It's led by Professor Stein. Basically, there are many other partners collaborating with different collection, core collection and bridging collection. The idea is collect all this data, test it in different environments, use the genotypic data for genomic prediction, and also create an infrastructure and a connection between the different gene banks across the world. And these are two new projects that we are pretty excited to start from next year, along with my colleague Miguel, to Horizon 2020, based on the study of the root system and their interaction with the soil. And this is the second one. I think some of the other partners are around. And thank you all for your attention.
Very interesting facilities you're building up there. We move on now to Niels Stang, please. And this is Gardersleben, the file collection at IPK.
Thanks, Søren. I will save us some time. I realize I should have maybe included a few aerial view pictures and all our facilities, but I recommend for those who want to see the facilities, come to Gardersleben and see it live. All right. IPK barley collections. I took a picture or a screenshot from the Eurisco website because IPK at the moment is also hosting in person of Stefan Weise, who is leading the Gene Bank working group on Gene Bank documentation. He is currently in charge of the Eurisco database, which is a resource run by the ECPGR, or please help me with the abbreviations. It's the office installed by the FAO. The Eurisco database is basically meant to collect genetic resource information from the different gene banks. And what you see here is just a list. I can see it. I didn't bring my reading glasses. But you see, if you query the Eurisco database for keywords, I queried for barley, you see a list of the different gene banks that have submitted their information with the passport information, and on top you see Germany with 21,600 whatever accessions. So IPK in Europe is the largest gene bank for crop plants and wild relatives in the EU 27, and barley is the second largest collection in our institute next to wheat. I think we are still at the moment the only barley collection that was genotyped as a whole in a project that was funded by our mother organization, the Leibniz Association. So we won a project in 2015 that was a pilot, really a conceptual pilot project to genotype an entire collection. And we choose barley because barley is an important crop for IPK as a model for many different purposes. But barley is also an inbreeding species, selfing species, highly homozygous usually. If you go into land races, of course, they can be heterogeneous. There are all these problems that you anyway find in these collections. But barley in this respect was definitely one of the easier species to start with. And what you see here is just a screenshot or a PCA analysis of that population. So genotyping an entire collection is very useful for doing science, but it's also very important information to improve the management. And this is something that will not happen at the moment when you really generate the data, but it is really information that has to be taken also to inform the stuff, because there is a lot of concerns. I talked during that project and also now during the agent project with so many different gene banks and their philosophy of running their collections, and if they would follow this approach or not. So one can spend the whole evening in discussing this. However, I want to make a point that we have genotyped a single plant from each of our 20,000 accessions. It is an approximation of the genotype of the gene bank accession because we only selected a single plant, but it is useful data. But what we did during that exercise, and this is even maybe more important, this is a PCA of a different view, overlaid with color codes of the so-called barley core collection. The barley core collection, I think, Janne, you have mentioned in your presentation as well, is a collection that was put together before molecular genomics by a number of well-known barley geneticists. And they basically put together a collection on the basis of geographic origin, of phenotypic variation, and so on. This was a great achievement, but tell me why this collection wasn't used. I think there are very few papers that really relied on that resource, and the most prominent complaint was always, there are accessions that are heterogeneous, we cannot work with it. But I think you have to go use the material with the right intention and then work with the difficulties. But what I want to show you here, the barley core collection, before molecular data was available, was a very good compilation of global barley diversity, because you see that all the subpopulation structure you can see in that PCA is also covered by red dots in this BCC. So I want to advertise this because the BCC is part of the living collection of IPK. You can go to our ordering system and you can order that material. But we also decided to use also the entire diversity information that we captured during this GenBank project. And that is shown here in this overview slide. We decided to subsample nested, what we call core collections, of 1,250 individuals, which we then dedicated to different resolution or coverage of resequencing. Not all of this data has yet been published, but will be published in hopefully within a year from now, as we are in the process of finishing the data generation for the second phase of the Pangenome project. The lower plots also show you just the material that we selected for the Pangenome for whole genome sequencing. So for the barley collection at IPK, there is not only the GBS data that was used or that was generated during the genotyping of the entire collection. We have also generated different level resolution resequencing data for selected material that was chosen to represent as best as possible the diversity within the collection. So if you're interested in that data for bilateral collaboration, we could also talk about this pre-publication access. So a few of these HORE accessions were already fully sequenced and the data is also published. And Martin has given an overview of the extension of the project. You see here again the PCA and the dots represent the material that was selected for whole genome sequencing. So over 50 accessions of the genetic resources selected based on diversity, global diversity, will be available not in too far future on the basis of a whole genome assembly based on PacBio data, chromosome scale. And in that effort, we also included part of our wild barleys. You see in the right panel, the PCA, including also the wild barleys we have at IPK. You see the red dots are the cluster of wild barleys. IPK wild barley collection is overrepresented for material from Israel and is not fully representative of the true natural range where wild barley exists. That is indicated in the upper panel is a representation of the different sites where Spontaneum exists with the population structure based on molecular data. So the world barley collection from Brian Stephenson is there much more representative of the true wild barley diversity. And we included, in fact, from both collections material also in the Barley Pangenome project. To sum up, the official barley collections at IPK you can access through our GenBank information system. It is a very basic interface and you have to love it, but you can order the material through that interface. And most of the 21,000 accessions you can order them, and this includes also the barley core collection. What is not yet an official IPK collection is what comes out of the project. So our core collections that we selected, they were SSD material, they are SSD material, and that is part of the complication if you start doing research on collections. The leader of the GenBank has to take decisions at one point if collections become part of the collection or not. And as long as this decision isn't made, these are unofficial collections. We can provide them on the same basis of the SMTA, but you cannot yet order them through the GenBank information system. I have listed there also the Barley Pangenome version 1 and 2 accessions, which we are in the process of multiplying. For the Pangenome version 1, I have to admit that we had included cultivars, which we are not allowed to directly forward. You will have to get the material from the respective sources, which are breeders, so you either buy the seeds or you get it through them. And I think with this, I'm at the end of my presentation. With the picture that Ronja assembled, it was a few years ago, we had grown the core 1000, and there was just beautiful variation in the field. Thank you.
We move on quickly, and the next speaker is Nestor from Zaragoza, and it's Mediterranean land races. Inobar presentation, so please.
Thank you, Soren. So we move for this presentation from germplasm banks to actually a research project which is heavily based on germplasm. This project is called CandyBar, utilization of local genetic diversity for studying barley adaptation to harsh environments. It is coordinated by Agostino Fricano from the CREA. He's not in the audience. Luigi Cattivelli, Alessandro Tondelli, Rafaela Battaglia are also involved. Myself, I'm Ernesto Igartua from CSIC, Spain. And these are the participating institutions, also AIM Shams University from Egypt, University of Sfax, and Kukulova University, and from Turkey, and University of Lleida, and Max Planck Institute, Ivana Costa, and Gustavo Slaffer also in the audience participate in the project in one or other mission. So, I'm going to talk only about one part of the project, the one which is based on the study of the Mediterranean core reference set. That's how we have called a set of about 1,200 georeferenced barley land races. They have been multiplied by single seed descent, and the seed was contributed by project partners and also very generously by ICARDA, who gave us about 600-700 lines, and from the IPK, from the Bridge webpage, we got about 230. We are multiplying, and then more data. Because this study will be enriched for environmental association. We heard before about environmental association today by another 1,700 accessions the data from those accessions coming from the work that Niels Stein just explained a few minutes ago. So what we are going to do, this is ongoing work. We have already genotype by sequencing markers for the accessions. We are developing a high-resolution gridded data set of bioclimatic descriptors. We want to identify loci associated to bioclimatic variables by environmental association. We are also starting to do US for several traits, also as shown by Andrea before. And we are carrying out targeted resequencing to unravel gene haplotypes that control phenology in Mediterranean germplasm by exome capture of several well-known phenology genes. And there is also some whole genome sequencing of selected accessions, basically comparing accessions coming from hot and cold places. Just to show some results, this is also a principal component analysis similar to the one you saw before, but narrower scale, and combining together the accessions of the Mediterranean core reference set and the accessions which data we got from IPK, and many thanks to Martin Mascher, by the way, who was very generous sharing even the raw data files. This is done with about 11,000 markers. So that's the structure of the first two principal components. And this is a population structure of about 14 groups that we have identified across the Mediterranean basin. A typical population structure analysis, you can see more at poster 48. There are many colors here. Some groups are really narrow in geographic scope. Other groups are quite large. So they mark success stories along the Mediterranean basin. There are many things you can talk about that, but not at this moment. This is the same graph as before, but here differentiating 2 rows and 6 rows. 6 rows in yellow, 2 rows in blue. Looks like a fish to me, I don't know to you. And you can see that the separation is not that drastic. You can see like a group here in the tail of the fish, which is yellow, shouldn't be there. If we look at it a bit closer, then we see that there are a couple of blue dots there, and actually there is one here which is very similar to the 6 rows. The 6 rows are all from the Caucasic countries, Russia, Ukraine, some from Turkey, the Balkans, and that blue dot is from Syria. Who knows? Could be. Well, related to. This is a multiplication field we had this year with 650 accessions in Zaragoza. And with that, we measure days to heading, EC traits, and we mapped the days to heading here on this map. And you can see by the shading that the late genotypes are in the north and the early genotypes are in the south, in the Middle East. That's something you can expect. If you do the same for plant height, you get pretty much the same graph. Actually, the correlation between the two traits was 0.73. This is something which is probably you should expect, but I didn't expect to find this large correlation. Actually, if you do this plot of days to heading versus plant height for 2 rows and 6 rows, you can see that the correlation is quite big. And if you look at the 14 groups identified by population structure, then the correlation for this quadratic regression is astonishing. So any comments about this? I'll be glad to hear after the talk. Actually, in each group, there is some dispersion, of course. So there is plant height quite diverse at each germplasm group, but the general trend is quite visible yet. Doing variance components division between populations, within populations for several traits indicates that heading and plant height are probably most related with adaptation in this Mediterranean environment because most of the variance is between groups, whereas for other traits like spike length or flag leaf area, the variance is mostly between populations. We did some GWAS, and to my surprise, because this is just one environment, small plots, but we found a couple of good hits in days to heading. One is FT1, the other one is very close to HV10, so that was also expected. We didn't find anything in plant height. As I said, it's a carryover of days to heading. And we found probably some hits for flag leaf area and second leaf area, which are not that related to each other. We are starting the environmental association, and this is just a table of the 14 groups, several climatic variables, which are, well, I like to call them agroclimatic variables because they are related to the growth cycle of each accession of the different groups. This has allowed us to find groups that belong in cold regions, groups that were developed in hot regions, and that is helping us to pinpoint accessions like Ivan Acosta, for instance. He tested some accessions from cold and hot places, looking for sensitivity to heat at flowering stage. And this will be done, this will be used for downstream analysis. And a very rough environmental association already gave us some possible hits. I think it's too early to believe in them, but looking at the average temperature during flowering time on the top, on the average temperature during grain filling period underneath, there are at least a couple of good candidates to look in further detail in the near future. And well, I just want to thank Prima again for the funding. And this work was mostly done by Francesc Montardit, Bruno Contreras, Ana Casas, Santiago Begueria, and myself. And then the collaborators, the coordinator of the project in CREA, Agostino Fricano, and all the fellows at CREA, Hakan Özkan from the University of Cukurova, and I don't want to forget Martin Mascher again for his help with data. Thank you.
We quickly move on to the next one, which is on Fluturia and from Sweden, but it's on the origin of Icelandic barley germplasm.
Okay, thank you. Yeah, so I'm actually in Sweden, but today we're gonna show you something about Iceland. And we took on the quest to find out where the Icelandic barley germplasm originates from, but I think we maybe have to add the question mark. And I'm first going to give the word to Ranna. He's the responsible for the barley breeding program in Iceland, and he's going to tell you a bit about the background first.
Yeah, let's dive into this. So you might think Iceland is a warm and happy place, but to your surprise, it's cold and miserable. But it's pretty cool. And I want you to focus on this figure I'm showing you here, where you see the northern hemisphere, and the orange line on the figure is the limit to where the 10 degrees centigrade limit of the warmest month, and you can see that it runs right across Iceland, and it's generally regarded as the limit of the habitable world. And if you just focus on the island, it's right there between Greenland and Norway. You can and you move it with your mind to the right, I guess. Yeah, and put it into Scandinavia. You can see that Iceland is well, that Scandinavia is pretty much warmer than Iceland in the summer. And I have some further data to explain this. We clearly have a raining season. It's all the time, and there we see that the average temperature for the island it just skims over the 10 degrees centigrade line. But also please notice that it doesn't go very far down. It's just the average temperature just drops below zero. So we have warm winters and cold summers. And there you can also see that we are sowing at the end of April. we are heading there right about now, and harvesting is late September, early October, and sometimes into November. So it's a really long growing season, and but nonetheless, the settlers of the island that came about 1,000 years ago, they did bring cereals with them, and they tried to cultivate their cereals as they would have done at home. And this continued until 1402 at the arrival of the plague. And plagues have a terrible effect on island communities, and killed quite a lot of people. And subsequently, the agriculture kind of changed and moved into more herding sheep. There is some archaeological evidence of possible cultivation after the plague, but none after the 18th century. Until the man you see on the picture, he reintroduced barley to Iceland in the early 20th century. So what happens is that there are no land races in Iceland, and it has never been adapted to the special situation or environmental situation in Iceland. So I'm going to show you a picture of barley in Iceland. This is, you can see the picture is taken from top of the plot combine, and you can see the difference of adaptation of the genotypes on the picture. Some are ready, some are still green, and that's quite typical. And if we wait any further to cut that, it will be blown away. This picture is taken almost 66 degree north. You wanted this one also? No, skip this one. This one. Okay, I'm going to give the word back to Flútor. I just had to nudge in here to talk about Iceland.
Okay, thank you. Yeah, so and then but there is a barley breeding program in Iceland, as I mentioned, and so the Icelandic ideotype is short and early, but it's not always what the farmers actually want, is it? No. Anyway, but crosses have been made with with land races from the Faroe Islands, with Danish dwarf genotypes, and with other Nordic material like Sweden and Norway. And what we did now is that Rana genotype 65 Icelandic breeding lines, and added 28 parental lines. And for the analysis, we added 19 potentially related genotypes that we took from the 1,000 genotypes from the barley core set. So that I'm going to call IceBar, or that's what Rana calls it, and it took me way longer than I want to admit what the bar stands for. And so, but we analyzed that set. It was about 112 lines, and then we added those lines to the 1,000 genotypes. And because everything is genotyped with a 50K chip. And so this is first the core set with the Icelandic lines, with the IceBar lines. And we can see a bit, like Martin mentioned on Monday, the north-south gradient on the second axis. And to the first, on the first axis, it's the eastern line genotypes. And the Icelandic genotypes cluster like in the upper left corner. It's like close to the Northern European lines, which we would expect, since we know that they were crossed with those lines. Yeah, but do you want to say something about that? And then I took only the 112 lines and did a structure analysis with it, and the highest peak was at K2 because it's 6 and 2 row barleys, and then there was a peak at K4, and then I looked into it a bit more. So we can see that so the Faroe Island lines, they seem to be like their own thing a bit, but we see some of it in Icelandic lines. Then there's the big group, the K4, which is a lot in the Icelandic lines and from K1 as well. And in the PCA plot, it's just, the colors correspond to the colors from the IceBar plot. Sorry, from the structure plot. So And to the right, we see like those purple ones, which seem to be mostly Icelandic lines. And yeah.
So I remember when Flutur was showing me these figures, and she said like, "Oh, there's nothing really important there. You know, it's just it's just a cloud of things." But I saw something completely different, and I saw a very clear pattern. And we were at the lab meeting with our third member, and I held a long talk about what I saw, and it became known as the Billy Madison theory. And I will dispense this theory now. So I want to say that the Icelandic barley is like furthest to the left, and I think it's pushing the frame to the left. And when we look further, is this the figure I want to show? This one? Yeah. So when we look at this, furthest to the left are the oldest elite breeding lines of the breeding program. And they are mixed in with Danish genotypes, breeding lines, and Scottish, and they're kind of like all over the place, and not really fixed in on anything. And then when we follow the graph to the right, what we're seeing is that that the lines are getting more elite, and they're getting younger and younger and younger until we are furthest to the right. Those are the most modern breeding lines and the cultivars that have recently been released. And they are going back to some super old European landraces and early cultivars, like from the 30s and 50s. So I think that's quite amazing. And I think when we look at this one, the youngest ones are like furthest down, and we think that this is the north-south gradient, so they're actually going south. So the selection and crossing has been trying to get adapted to Europe rather than Northern Europe.
Yes, and that's usually when I say to Jana that everything he said is just is one of the most insanely idiotic things I've ever heard. At no point did his rambling, incoherent response correspond to even close anything that could be considered a rational thought, and everyone in this room is dumber than they were before. And and this is the last slide we're going to show you, and then it's time almost for beer, I think. It's just the nice genetics distance for this, and we see a bit the four clusters, but it does not 100% correspond to Rana's mad theory. Thank you.
And we move to the last presentation, which is from Australia. So we get almost all the way around the world. And this Changdao Li, Murdoch University, barley germplasm in Australia.
Thank you. Now we are from Northern Pole to the other end of the Earth, so from very cold place and to very hot continent. Now, the very interesting is yesterday, actually, someone in the audience asked me, are there any barley germplasm in Australia? If you have the same thought, you're partially right. Actually, in Australia, there's no wild barley, even no landrace barley in Australia. Now, Australian barley actually has very clear and short history. The first barley brought in Australia is the first British settlers about 200 years ago, and we still have that variety called Chevalier. But the European varieties have been struggling in Australian hot, dry continent. In the first 100 years, they simply couldn't make the barley growing in Australian continent. So that is coming later. We said germplasm is so important, and Australia have used the European germplasm as the backbone, then cross with a variety from other places in the world, especially from the North Africans. Then now we successfully transferred Australia to the world's largest multi-body exporters just in last 60 or 70 years. So today I just, but we, even though we said that there is no germplasm origin from Australia, but we do collect it and quite a lot of germplasm. Now, the first major germplasm collection in Australia is obviously the national facility in Australia Green and Gene Bank. So this is located in the Horsham of Victoria. Now, this is a new facility. This only opened in 2014. Okay, and they claim this is a very innovative design with double skin freezers designed. So that means the germplasm can maintain there for under minus 20 degrees. The facility initially designed for collecting the Australian native and species, but not also the major facility for like crop. So at this stage, and then they collected about 20,000 different body genotypes, certainly, and that include all Australian varieties, but the majority of them coming from Europe and Central Asia regions. So this one, it is actually one major Australian body germplasm storage, collection, and distribution centers. Journalists can provide small amount of seeds for the research purpose. Then we spread into the different places around Australia and doing germplasm characterizations. So one such research project is currently in the University of Adelaide, and they call Oz Barley project. Now, what's the Oz Barley project? The journalists have about 400 all Australian barley variety and key and breeding parents. So journalists, they have to generate the full set, the phenotype data. Now, I'm currently also working together with them, going to generate the high coverage genotype data for this for around 400 the key variety and germplasm in Australia. So that means pretty soon, if you're interested in Australian barley germplasm, you'll not only have the genotype data, you also have all the phenotype data and for your interest. The other major germplasm activities, one is for disease. One is in Sydney University of Sydney, that's Sarah Russ Research Centers. Journalists in that center is the major characterization barley germplasm. Certainly they also collect other cereal germplasm major for different rust resistance. So if you're interested in the leaf rust resistance for various rust, so the Sydney University and the Sierra Rust Research Center, obviously that's your contact point. Then the other the disease centers do the other disease, and that is called Center for Crop Disease Management. That's the Curtin University, and that's at Perth, at the same city as I am. And so they're doing the germplasm characterization for all the other barley disease, like gnatform, gnat blotch, spot form, gnat blotch, and podomildews, that kind of disease. And then the other one, the major activity, I think, from the University of Tasmania, then they were major due to germplasm characterization on the barley waterlogging tolerance. So if you are interested in waterlogging, the research in the University of Tasmania, and it is the place. Now, last fall, we got into my own group. So we call ourselves Western Crop Genetics Alliance. It is a joint research centers between Murdoch University and the Western Australian Department of Agriculture. We have collected about 6,000 barley accessions from around 41 different countries. And our major interest in it is the phenology development and the germplasm adaptation to the Australian continent. And we based on that is the passport data, phenotype data, then we build up the one about and 1009, we call it our core collections. So we generated a low coverage and genome sequence. Then we use this low coverage genome sequence for about 1,000 lines. We identified some of the key genes for the barleys to adapt to Australian environment from the cooler European continent to Australia hot and dry environment. Now, the other major things we're doing in my group is we have a large body double haploid collections. Currently, we collect about 5,000 and double haploid lines. So this includes a lot of, and the mapping population have generated in Australia. Now, for these materials, there's a lot of cross, not only in between Australian varieties, also have the cross from Australian variety with the European variety, and Australian variety with the North American and varieties. So this can be most of the populations or they have the full genetic map available. So if that is someone interested in and looking understand the genetic difference between Australia and Europe or between Australia and North American, there is some type of available. Then we collected about 1,000 the Bowman back cross lines that Jerry brought into Australia when he was the body breeder in Australia. So obviously currently we're interested in to sequence some that is Bowman back cross line for various and mutant genes. And the other things we have done regarding germplasm, we have a collection of arched planets and mutant populations. Another is the Vlaming populations. Now, finally, so one major thing we have done in our group, we have done large scales and also soil screening. So we have screened a lot and also soil tolerant germplasm. This one actually is a very successful story regarding Australia. If you're looking at the map, so that is the part. Generally, all the green colors is acid source, yellow is the very heavy acid source. You can see the majority our green bells is acid source. So we screen germplasm and to identify the markers of genes that develop on the commercial varieties. So we finish the whole pipeline. So if anyone you are interested in acid source and tolerance, you can contact us. Thank you for your attention.
So hello everyone. For me, it's like a closing session. So it's an honor to be at the closing session today. And my name is Aigars, and I'm from a small craft brewery called Valmiera Muiža. It's a little bit strange name. But when you will practice, you will see it. So basically, it's Valmiera, and Muiža means like manor, like chate. Yeah. So it's Valmiera chate because every perfect beer needs perfect manor, so also our small brewery is brewed in the old historical manor of Valmiera Muiža, which is a small manor some 100 kilometers north from Latvia, between Riga and Estonia, like Tartu on the way. And today my idea was shortly to tell and present some tastes, because you have a lot of passion for barley, and also we have a lot of passion for malted barley, let's say. And that's why we will move to beer tasting. We will have jointly, there will be 4 different tastes today. And the first taste, which we will present to those who can enjoy beer, will be our most popular beer type, which will be our Amber Lager. This one. Then the second will be smoked beer, which will be smoked barley. And I will tell a bit more about this. And the third will be Riga Porter, Baltic Porter. So if you are in Riga, you must try Porter, because this is also something very special for Latvia and Riga. And those who cannot, for different reasons, enjoy alcoholic beer, we will have kvass. This is a strange drink from Eastern Europe, as you know. Yeah, so this is kvass. Basically, it's a very low fermented rye barley malt drink. And it's non-alcoholic, because the alcohol is around like 0.5% only. Yeah, so it's basically non-alcoholic. And very, very shortly about our brewery, and a little bit shortly about our philosophy of brewery. So the idea for our brewery is very, very simple. As you know, the national drink of French people are wine, probably. And when they celebrate with champagne, the national drink of Scotland is whiskey. Probably everyone knows it. The national drink of Latvia is. It should be, yes. But what I see when Latvians celebrate, at least it was 10 years ago, when Latvians celebrate, they actually on the table had French national drink and they had Irish national drink. And I didn't like it. So I said, we need that Latvian beer we take back on festive table, because beer is a Latvian festive drink. Because festive drink means you put it on table historically on the most famous, most important moments of life. Which could be the most important moment of life? It's, for example, getting married. So historically, always there should be beer on table. The same like also everyone maybe has heard in Latvia about Midsummer Eve celebration, like we are also with Lithuanians the last pagans of Europe. So we still celebrate in midsummer. No one goes sleeping, everyone makes fire and everyone drinks beer, because this is also one part of tradition to celebrate midsummer, which means when the nature has the best like fertility, and then we celebrate the shortest night. And of course drink beer, because this is given us as a gift from nature and sun. So that's why my idea was to make beer back on festive table, to make it the same like wine. Because when we talk about wine, we always say, we enjoy wine with nice snacks in the restaurant. And when we talk about beer, I drink beer, eat chips, and watch TV football. Like something like this. But I think if it's celebration drink, we say in Latvia, we never drink beer in Valmiera Mūža, we enjoy beer. So today we will enjoy beer, no drinking, yeah, enjoying. The first taste which we will enjoy will be our amber lager. So basically it's lager, as you know, so which means it's bottom fermented. And as you know, also Latvia, after 19th century, became like a lager country, because the manners started to build very modern breweries, and lager became very popular and substituted ale type of beers because of very nice malty taste. And how this beer will be very different from what you maybe know in Germany and other countries. This will be very mouthful, so it will be more rich in taste. So what we say, it's not beer that you should drink a lot, but you really more enjoy it with some food. So it's more malty, more mouthful, and the taste will be very balanced. So it will be malt and hops in a very nice balance. So it will be not like that hops are over-dominating. So it's not like IPA, you get like hop like. But for you, like people who are in barley business and studies, I think here you will be enjoying 3 different barley varieties in the taste. So there will be one barley, which is a Pilsner malt. When you will be going and enjoying, you will also be able to see these malts. And we have here Munich malt, which is slightly more roasted malt. And we will have also caramel malt. So 3 malts in combination, so we get more mouthful taste. Probably you know beers, why they have those strange names for these malts. Because Pilsner malt is like the malt type inherited from Pilsen when they started to make these very pale color beers in 19th century. So now everyone in Europe, everyone globally calls this malt the lightest malt like Pilsner malt, which gives a sweetness to our beer. Munich malt, so this gives more mouthfulness. So all the malts which are slightly more roasted, slightly darker in color, are called Munich malts, and we use also this for mouthfulness in our amber lager. And we use also light caramel malt, so the beer gets at the very end a little bit touch of light caramel, so then it gets a little bit more mouthful. These malts we do not produce ourselves because, as you know, malting is a very big and complicated business. So we buy malt from 2 German malteries, which is Best Malts and Tirex. And also we buy from Latvian maltery Latraps for Kvass malt, which we will talk later. And some of you already get first glasses of beer, and I will tell you how you should approach beer. Actually, I should not tell you, because everyone enjoy wine. So the same way you enjoy also beer. So the same like for a wine, each special flavor deserves special glass, because the idea of glass is not just to look nice. So the idea of glass is to emphasize the taste. And for our beer, we decided that this glass will be the best for our taste. So it's like a tulip form of glass. And the same like for wine, you start to enjoy with some ice. With some ice, we start enjoying. No problem. I'm just joking, yes, so don't worry. So we start to enjoy with some ice. Yeah? We look to the color. And what makes this beer special and stand out from like pale beers, that it's like amber colored. And this amber color is given by this Munich malt, which is slightly more roasted malt. And this also gives this mouthfulness to this beer. And if we talk also about lagers, it should have a nice foam. So if the beer has not foam, probably there's a fermentation defect. Yeah? So that's why it's always good to have a foam. And then you could see, is it filtered or unfiltered? What you see, this is a filtered beer, because you see it's perfect transparent, like a polished amber. If it will be like cloudy, then it would be probably unfiltered beer. So this is a filtered beer. And what we see is a nice bubbles inside, which means it's like for champagne, that it's coming from natural fermentation. So it's not like artificial added CO2, but comes from natural fermentation, the same like our champagne. Basically, it's champagne, barley champagne, basically. Yeah? Yeah, so that's why we call it Latvian champagne on festive table. And then next we start with aroma. So basically the same like with wine. You try, and when you try it, the same like with wine, you need to use both these. Like it's like a dog basically. They are experts in smelling. Yeah, so look from the dogs, and you will see how to get the best aroma. And for this beer, to create our aroma, you will get some sweetness, nice honey sweetness. Of course, it comes from the Pilsener malt, this nice honey sweetness. A little bit also of some deeper honey notes or caramel notes comes from this caramel malt. And you will get also some hop aroma. We use noble hops from Hallertau region called Hallertau Mittelfruh in Germany, because in Latvia, unfortunately, there is too little sun to grow hops. We see there is possibility to develop special barley varieties to grow even in Iceland, but probably varieties will help for Iceland to grow hops, because hops really need a lot of sun. So actually the hop growing could start in Lithuania and maybe even and then south from Lithuania. So we are unlucky. So in a way, so we have to import the hops. So and then when you enjoy and take the first sip, so the whole tongue is activated. And what is also special about beer, when you make wine tasting, after the sip you throw it out. You know, it's like spill it out, spit it out, spit out. In English, just spit it out. But in beer, you should not do like this, because beer has also aftertaste. And aftertaste comes from hops, so you always need to swallow it. So that makes beer also more special. And the same like any drink, when you enjoy it, the beer has this first taste, you get a little bit this freshness, then it's a middle body taste, and the long aftertaste. So beer has these three different tastes. If you make it nice, then you get this sensation like in 3 steps. So everyone has, okay, you have to wait a little bit. Yeah. And then I will use this break while everyone gets some beer. I heard that someone has birthday today, and as you know, beer is for festive table, and that is Latvian champagne, barley champagne from Valmiera Muiža. It's the same beer, but in a nice bottle. So there are 2 options. One option is you enjoy it today because it's your birthday, or second option, you wait till December 31 midnight, and then you wait the new year with this. Yeah. So just to change perception of beer, because I think you also, I'm so excited about all these barley discussions, because also in the breweries lately, it's also interesting not only to make special hop varieties, because as you know, the craft beer business is very much built about hops. You make such hops from USA, from New Zealand, bitter, citrus, a lot of hype about hops. But we, as in Valmiera Muiža, we like really to base our beers also on malt, especially lager beers. It's good to base on malt, and that's why we also are interested in specialty, how we could get more special taste from barley. One, of course, is through malting process. because there is a lot of the malteries are the kings basically. But another which becomes more in a way trendy now, the same like for wine, is that you start to use very special malt varieties. And we have made some experiments with some Barke variety from Weyermann, which is patented varieties, and we get some very special taste. We have tested also some malt which is made from Pilsener, like floor malted Pilsener malt, which is in Czech Republic, some more special malts, and you get a little bit more interesting, more special tastes. So me as a small brewery, I'm truly interested that if there are some special malt varieties for beer, which in combination with nice malt and a special barley variety, we could get some more specific tastes. So this is very, very interesting. So if later on I will show my contact, so if someone maybe has some, but we do not have a maltery, so which means that there should be a maltery who helps us to make this malt, and then we could brew some beer and experiment with different malt varieties. So this is, and especially if it's something like Nordic, then it could be absolutely excellent. If to find some special new taste varieties, and for us as breweries, the amount of harvest is not important. For us, it's important the taste. So how much more taste we could get, not how much more tons we could get. So this is the challenge. And this is quite different from what normally used like barley for feeding cattle. Or so that's why this is a very interesting direction. So everyone has the first beer, or we are okay. We are rushing. Yeah. So you are quite many. So that's why we will carry it around. So then I will talk about. Quick question. Do you have these ones? Like I know that we will have some snacks also today made, but I do not see the snacks. Okay, so it will be self-service. Here on this corner, there will be some special. You will walk around. So I will, while everyone gets a glass of beer, I have an honor also to present by one small, I understand, home producer, special barley, we could call like cakes or pancakes. And there will be 3 different varieties. What is special, this will be made from Hules barley. And it will be made from a Latvian special barley variety, which is called Cornelia, Cornelia barley. And there will be one which is with honey. The ingredients is wheat flour, honey, butter, kefir, salt, soda. Second will be pea with pea scones, Hules barley, Cornelia flour, grey peas, onions, smoked meat, salt. So this probably you will have to taste with our smoked beer. So probably it will be a good combination, smoked beer and with some smoked meat pancakes. And the third probably will be the favorite for last. It is cannabis scones. And it will be also Hules barley, Cornelia flour, cannabis, sugar, salts, plant oil, garlic, water. Yeah, so nice enhancement to beer flavor. It could be also, I think, for last, for dessert. So also to show, because when I was a kid in Latvia, very often when you go to shop, there was possibility to buy barley bread. Unfortunately, after Soviet time, it's somewhere we lost this very small bread, like round small bread, which was warm, and we eat it a lot of barley bread. Somehow today it's lost, but maybe this will be a good inspiration for some of you. So then the next, what is also important when you enjoy beer, I'm just trying a little bit to keep up while you everyone get a beer. So it's important temperature is for beer. It's the same like for wine. So if it's amber lager like this, which is quite light in color, the best temperature is 5 degrees Celsius. Why not to drink it colder? You know that you drink cold bad Russian vodka only, because then you don't feel taste. So if you don't want to feel taste, you make it extra cold. But if there is something you want to feel taste, drink it warmer, enjoy it warmer. So for this beer, the best temperature will be 5 degrees Celsius. The next beers will be darker in color, more malty. This will be served and best enjoyed a little bit warmer. So each beer also, to get the best enjoyment, it's important to get the best temperature. It's the same like wine, basically. And normally craft breweries will write on the label which is the best temperature for enjoying beer. The next question sometimes that people ask, how many beers you should drink in one evening? Not to show up to tomorrow, you will show up, but actually it's only 3 beers you will enjoy taste. So that's why today it's only 3 beers. Because with the 4th beer, actually you lose possibility to feel a taste to the best. The same is with wine. So that's why my recommendation is, if you go to expensive restaurant or to shop with some craft beers, do not save on first 3 beers. Save on the next ones. Yeah, because on the first 3 beers, it's always good to enjoy them. But it doesn't matter actually. In a way, the quality you will not feel so much taste when you drink the 4th or 5th or 6th beer. So the best, of course, is 2 beers. Yeah? or 3 beers. Yeah? The next question sometimes that people ask, what is the best glass for a beer? Is it a 0.3, the small glass, or 0.5, like this glass? What is best glass for beer? 0.5? The bigger, the better, yes? It's like in Oktoberfest, it's 1 liter, smallest. Yeah? Actually, it's in a way, it depends on temperature outside. Like if it's a hot summer, I would always suggest a smaller glass, the better, because it warms up very quickly. And I think it's always important that the first sip and last sip is as good. If it's too big glass like this, you for half an hour will be enjoying it, then the last sip will be very, very warm. And also without CO2. So actually the last sip will be the worst. So that's why I recommend in hot summers to order a small glass and then order another one. Because again, you will get a fresh one. Because otherwise it gets too warm. But it's again, if you want really to get the best enjoyment out of, from the beer. So, those who got the beer, some comments, some questions so far? So the principle when we brew this very classical type of beer, because this is very classical type of beer, so to really, we say like to put it back on festive table, it has only 4 classical ingredients. It's like this, like beer purity law, if you want to call it like this, yeah? So it's water, which we take from our deep well, then it's of course barley malt, then it's hops, and it's yeast. And for this beer, it takes 1 month to brew. Normally 1 week we ferment, and then 3 weeks we make this slow after fermentation. And how it's also different from the very big breweries, we do not pasteurize it. So it's unpasteurized beer, so it always keeps up the best taste, because pasteurization makes the taste a bit more flat. Yeah. So normally, if you do the work correctly, you do not have to add any artificial CO2. All this CO2 is generated in the process of fermentation. When the yeast eats the malt, the sweetness in the malt, it generates alcohol and CO2, same like champagne. And then for the brewer, it's important on the right time to close these valves, so to keep this CO2 inside. If there has been some problems in fermentation, then sometimes they add artificial CO2. But normally, but from my experience, I see if you add it, you lose a little bit of a nice taste, because if it's natural CO2, it has these very nice silky, the same like champagne bubbles. But if it's artificial CO2 added, it will have like Coca-Cola bubbles, the big one and rough one. So if you want this very smooth, silky, soft taste, but we always aim as a small craft brewer, it's always important to really get this CO2 from natural fermentation, not add from artificial CO2. The only where we CO2 use is in the filling process. But in filling process, it's where we need it, because the beer is foaming, so you normally make vacuum in the bottle, add with CO2 to create contact pressure for beer, you fill it, and then this artificial CO2 goes out. So it is only for filling process necessary, but normally there should be no artificial CO2 inside, if you do it in a craft, slow, patient manner. So if you are in a hurry, then of course you could always save on these steps and make it cheaper, but it will be on the expense of taste. So that's why we do not do it as a small craft brewery, because we brew only like 1% of all Latvian beer in Latvia. So it's we are like small ones. So we focus everything on taste. So to give best beer for restaurants. Good question is, when we developed our recipe, we experimented with a lot of yeasts. And we found one yeast which is special for our beer. It's not cultivated unique by us, but it is taken from one yeast bank. And then we cultivate this yeast. And normally brewers don't like to tell which is a yeast, because they say it's a small secret. But in today's, everyone of course can find somehow it, but normally we say it's yeast, it's lager yeast, it's special lager yeast, but we don't like to loudly tell what is exactly this. We say you have to find out. It's a pleasure of going this journey of finding out different yeast properties, and you could brew your own beer at the end. This is for our lager beers. This is for champagne. In our brewing process, we don't add any sugar before bottling. So it's no, because this all CO2 comes from natural fermentation, and we have the filling equipment now where we can create counter pressure in order to fill it in the bottle. So we don't need to make this CO2 adding inside the bottle. So it's no needed. No sugar. So for this classical amber lager and the next, which will be smoked lager, it's just 4 ingredients. It's very, very clean thing. It's water, barley, hops, yeast. For the very last beer, which we'll enjoy, and for the kvass, there is some sugar added, but it's a little bit different story and different way we do it. But for the first 2 beers, amber lager and smoked lager, there is no sugar added. It depends on brewer. So we say you add sugar if you so it's the most easy way it's easier way to brew, let's call it. Because to find the correct barleys and barley combinations, and malted barley is more expensive than sugar. So that's why sometimes you add, because it's faster, it's easier, it's cheaper, it's more simple. Or there should be some historical reason, like for kvass or for this porter, which we will enjoy at the end. But for very classical beers, we don't do it. Except in some beer cocktails. If you make some radler or shandy, then of course you mix beer with lemonade, and lemonade has some sugar inside, of course. But normally we say you should not add any sugar. You get all the sweetness from barley and all the CO2 from natural process of fermentation of barley malt. So actually you don't need to add any sugar. And for us it is a challenge to brew without sugar. Normally these Valmiermuiža beers. It's more difficult, but it's more exciting. It's more pure stuff. It's more like historical stuff, like in old days. So then I will move to the second beer, which will be also some look into history of Latvian and Nordic brewing. It will be the dark smoked beer. And why we call it smoked beer? So probably everyone knows about peated whiskey. So peated whiskey, like in old days in Scotland, you have peat, you need to dry out barley, you use this peat to dry out, so barley malt gets some peated flavor. Actually, the same happened also with beer in brewing for Latvia. In the old times, we had these black saunas, and in these black saunas, on the top of black sauna, we also were drying out barley malt. Because this was the most energy efficient way. You go to sauna, you take some smoke out, then you were smoking fish, you were smoking meat, and you were also smoking this barley, because the barley you need somehow to dry it out to get this malt flavor. And then we recreated one flavor with this smoked malt, and in this beer it will be only 20% of this smoked barley malt. And then when you will enjoy it, you will get a little bit of taste of this. Some will say it's like a smoked ham, some it's like a smoked sausage, but it all comes from smoked malt. And you could just imagine how maybe 200 years ago or 300 years beer was tasting if it would be 100% of smoked malt, because this was how historically actually beer was tasting, very smoked. But if you enjoyed it in historical times from the childhood, it seemed to be a normal taste. But today already we say this is already not normal, because we say it's too smoked. And this probably will be very nice with these pancakes, which contain a little bit of this ham inside. And then this barley we smoke in birch smoke. So which is also a specialty for Latvia, it's birch smoke. To give a little bit of entertainment for you, there will be smoked malt, and I will give around this. You could just smell it, because you will a little bit get of this sauna feeling from the malt. if someone could also eat it, but better smell it. Yeah? So you could give around and just you will get a little bit of this special malt feeling, which comes from small smoke. And this dark smoked beer, it will be in color slightly darker because we use also a little bit more roasted malts. So it's easy to see that it will be like very deep amber color. So on the left side, it's this amber lager is the first which we enjoy. On here on the other side, I have this filtered smoked dark beer because it has a dark amber color. And while some of you are getting introduced to this dark smoked beer, I will explain to those who are not enjoying alcohol today, which is Valmiera Muiža kvass. And for this kvass, there are two things interesting to tell. Of course, first, it's non-alcoholic, but second is we use here Latvian barley malt from Lat malt or Latraps, which you see this advertising outside. There are these bags. So it is from Latvian barley. And second, what makes it special, we use also rye malt. And this rye malt comes from very small historical brewery, which still makes this rye malt like 100 years ago on the floor, fermented, floor malted rye malt from Nordics of Latvia, from north of Latvia, from Naukšēni, where they grow rye and where they also malt this rye. And then we get a lot of this dark rye bread taste, because in order to create a high quality kvass, as we like it, like in childhood, we used to enjoy, you need to get this dark rye bread feeling in the kvass. And then kvass also gets a little bit of sweetness or sugar, because this is like historical ways, so it was made, so normally they added sugar to get more fermentation. So this will be the kvass for those, and actually everyone could enjoy it optionally as a taste, the second, the third, or the fourth taste, because then you will be introduced to this so-called, like in a way, historically non-alcoholic beer malt drink. Because when I was a kid, I was not allowed to drink beer. I was allowed always to drink this kvass. And for us, it's always a challenge to create a taste which is not so sour, which is not so sweet, but you really get this taste of dark rye bread. So this is for this kvass, which will be also as option for those who. Then you ask our helpers, and they will serve you also kvass as a nice dessert. And then I will take one glass for this beer, and I will present the third one. So the third beer taste. The beer taste will be a beer style which has originated at the Baltic seaside. But this is a world-famous beer style. So if we talk about Czech-style beer, everyone knows Czech-style Pilseners. If we talk about German-style beers, everyone knows Munich type of beers. We talk a lot about Belgium-style beers, everyone knows. But at Baltic Sea originated a special beer style which is called Baltic Porter. And now the Baltic Porter is brewed worldwide by craft breweries in Canada, in USA, of course also in Baltics. And why Baltic Porter is special, and why if you are in Latvia at the Baltic seaside you should try it, because the Baltic Porter was very much enjoyed by royal people before the World War I. And somehow local brewers in Latvia mostly were Germans. And then they decided to take this British porter recipe, which is an ale recipe, and made it into lager recipe. So British make porter with ale yeast, as you know, normally in London porter style. But they started to make it in the at the Baltic seaside, in the breweries, porter with lager yeast. And as a result, you get a little bit more malty taste. And when we investigated, even we saw that in the beginning of last century, there were special porter houses created in Riga. And in these porter houses, the porter was served the same like coffee is served. So you could add some sugar, cream, cakes, mix in cocktails, and etc. So that's why this beer is slightly sweetened with caramelized sugar. Just a little bit to remind about this time. And it's like a dedication to Riga, because Riga, as you know, is famous with Art Nouveau as an architecture. Some say that Riga is like the capital of European Art Nouveau. But at least one of the cities which is rich with this Art Nouveau architecture, and that's why the design is also created with this Art Nouveau. And so Riga is famous with porters historically, and Riga is famous with Art Nouveau, and so we decided to combine these two things and create this special recipe, which is Baltic Porter. And this is the most dark and the most roasted beer. And in color, you will see it's really like coffee. And why and also it tastes a little bit like coffee, because here already the malt is roasted the same like coffee is roasted. So that's why it also gets a little bit of like coffee bean like look. And I will give this also around. If some of you wish, you could a little bit try it. So you will get also this coffee roasted like taste. So that's what makes this porter. It's sweet and it has this coffee like flavor in it, and it's perfect dessert beer. So the beer could be also like a dessert with some cakes. Today we don't have any chocolate cake, but next time you are in some cafe, ask for porter, and instead of coffee, order porter and enjoy with some chocolate cake as a dessert beer. So basically, 3 different flavors. So we start with amber lager, which normally is like an aperitif beer. Then we go to this dark smoked beer. And at the end, as a dessert beer, best could be this Baltic Porter. And also, if you make some beer tasting, it's also good always to start with some lower alcohol, lighter color beers, and go to higher alcohol and darker beers. So with each next beer, you could get the best enjoyment. So the tastes do not conflict or compete with each other.
So we also have porters.
I think in Finland, I know this Sine Bruhof is making Baltic Porter. About other breweries, most likely, most of them will be also this Baltic Porter. Because I think in Finland is also like a lager country, we could call it. Yeah? So I have seen only from this brewery some porters, but maybe there are some new breweries popping up all the time, and maybe they also make some porters. And if you make the porter with extremely high alcohol, like maybe some 9%, then you add name Imperial. Because historically it was stories that Russian royal court enjoyed these imperial porters instead of champagne. So it was like something like very festive, very special drink. Yeah. So we have Baltic Porter. It's only it's 6% alcohol without imperial, because we want that it's still enjoyable and drinkable, because 9% is already like a dessert in a small glasses. Maybe you could enjoy it only. Yeah. Yes, so what you tell, yes, that this special roasted malt could be used also as a coffee substitute. It's like a barley coffee. And actually, in Soviet times, when I was in kindergarten, they gave me this coffee. And you should not do it, because you will hate barley for life afterwards. Yeah, so that's why it was not very good experience. So, but in beer, I like barley. But in coffee, I don't like barley. Yeah, but yes. So it was like a cheap coffee for kids in kindergarten or school. So, but now I know that there are some craft companies, like very small artisan companies. They try to recreate these barley coffee-like flavors, mixing with some other. You mix some chicory, barley, roasted barley, and add maybe something else inside, and then you get quite interesting taste. So chicory and roasted barley goes very nice together. So there is some new development to make it also like in a nice and enjoyable, exciting way. So, and if some of you, we will have to use tomorrow Riga Airport, then of course you could also take find our beers in Riga Airport, because we more and more want to show the beer culture, and also in the Riga Airport you could buy some of our beer. And if some of you will be VIPs and you will take Air Baltic business class, then you will be also getting a free compliment beer. But I have never flew business class because it's too expensive for me. But they promised me they will serve it. Yeah. So if someone flies, you could tell. Was you able to get also a glass of Valmiera Moja beer? Some question, it may be also when you have to choose beer, unpasteurized beer, why you should have it in a dark beer glasses, like glass bottle, in dark beer bottle. Because for the beer, it's the same like for wine, so it is like sunglasses for beer, because light is not very good for beer taste. So you always, if you buy beer, better keep it in a dark place. Corona is pasteurized beer, and maybe for this pasteurized beer, it's no problem. And they also use not only barley malts, they also use corn and maize sometimes. And maybe this also is a little bit different. But for this beer, for our beer, unpasteurized beer, if it's on the sunlight, we see the taste starts to change, not to the pleasant direction. So that's why it's best beer to keep always cold and dark. So this is the best for a fresh, for unpasteurized beer. So this is unpasteurized, but if it's unfiltered beer, which means with a yeast inside, so the beer could be filtered and unfiltered. If it's unfiltered, there'll be a lot of yeast inside, and normally this beer even more requires some for lager, filtered lager. It really requires cold and dark storage, and it will have normally very short shelf life, maybe 1 month, maybe 2 months for lager. So if someone wants some extra beers, then you could approach bar, and we have some beer extra we could also add if you enjoyed some of the beer. So maybe we could make some quick voting from those who enjoyed all 3 beers. Everyone tried all 3 beers?
No.
2 beers? Okay, you still wait for the 3rd beers. Okay. So for porter. So for Baltic Porter, for this beer, we still have to be patient, and you still have time. So to also wait for the 3rd beer, the dessert beer. Yeah. So now it's step by step coming, because we have 3 people helping. So then I will help to pour beer. So if you have some more questions, you could come and approach me individually. So I think you have a lot of things also you can discuss between yourself. And how you can find me is, I have, probably you could ask the organizers, they will have my email, because basically it's my name, aigars@valmiermuiža.lv. So this is, if someone has some idea for cooperation, sometimes we make a lot of crazy experiments as a small brewery. So maybe, who knows, we could make some experiment with some of you, or with some of you know who could be interested to make experiment. And hope to see one day you in Valmiera Muiža and enjoy some beer in our beer garden. So, but this will be for next time. So thank you so much, and I will be here around. If some questions, you can approach me.